Why, despite years of research, are we not seeing the expected advancements in aging research? Why does calorie restriction remain the most effective yet species-limited and unproven in humans method for life extension? The answer lies at the heart of the intricate system of aging, a system with an enormous resilience. Aging is a complex process that occurs at a molecular level. Researchers are on a quest to identify individual aging genes, regulating molecules such as RNA, proteins, and other metabolites. However, the system of aging is not easily disrupted. If we alter the influence on a single factor or several related factors, the system perceives it as a minor disturbance, easily overcoming it. This explains why attempts to slow down aging by manipulating individual factors have proven to be ineffective. The solution, we believe, lies in cellular technologies. To achieve a comprehensive rejuvenation network, it's sufficient to replace old stem cells, which produce even older specialized cells, with young ones. However, why haven't cellular technologies, despite showing a reliable positive effect, pushed organisms beyond their natural lifespan? The majority of these interventions involved young but allogenic cells, that is, cells not native to the organism. Foreign cells, if they do settle in a new organism, do so only briefly. More often than not, they fail to settle at all. They are destroyed by the immune system and primarily exert their effect through young cellular signaling, a communication method used by cells which spreads throughout the organism upon the destruction of foreign young cells. When we introduce a lot of young signaling into an old organism, the old cells function more actively, but their age remains unchanged. They deplete their lifespan more rapidly, which may even result in a reduced lifespan for the organism. However, the quality of life always improves, as observed in one of Harold Katcher's studies. Even if we somehow remove the aging environment, for instance, through apheresis, the production of young signaling increases. The cells function better, but they remain old. Furthermore, in an old organism, cells interact with the old matrix through mechanical receptors, which is another variant of aging signaling. How can we overcome these hurdles? We need to obtain our own young stem cells and distribute them throughout the organism, allowing them to gradually replace the aged cells. For these cells to function as young cells should, we need to create young conditions. This involves removing aging signaling and adding young signaling alongside the introduction of cells. This method should lead to actual rejuvenation, initially in internal processes. After 10 to 12 years, young fibroblasts will produce a young matrix, replacing the old matrix that naturally disintegrates. This is when eternal youth should be achieved. In summary, while current research has not yielded the expected results due to the resilience of the aging system, the potential of cellular technologies is promising. The first picture shows a youthful organism. There are many young stem cells, youthful specialized cells, a youthful matrix, and youthful signaling pathways. The second image depicts a grown organism aged between 20 and 25 years. The specialized cells are still quite young. There is minimal matrix crosslinks, and slightly fewer stem cells. All tissues and organs are in optimal condition. There is a significant presence of youthful signaling pathways and minimal signs of aging. The third image illustrates a mature organism at 40 years of age. There are more matrix crosslinks visible, and the number of stem cells is slightly lower. The balance between youthful and aging signaling pathways is maintained, but there are initial signs of age-related health issues starting to develop. The fourth image portrays an elderly organism, aged between 70 to 80 years. There's a limited presence of stem cells, and the specialized cells have aged significantly. Aging signaling pathways dominate, while the production of youthful signaling is suppressed. The matrix has become rigid with numerous crosslinks, and age-related diseases are actively progressing. The next image depicts the removal of aging signaling through therapeutic plasma exchange. In the subsequent image, the administration of youthful factors, employing stem cells and exosomes, is demonstrated. In this image, we demonstrate the administration of our autologous pluripotent cells, whose generation we have successfully mastered. These cells serve to replenish our reservoir of tissue-specific, young stem cells. Over the course of about 10 years within an aging organism, young stem cells, under the influence of youthful signaling, generate youthful specialized cells. These cells begin to function as they did in youth. The aged matrix undergoes rejuvenation, transitioning to a more youthful state. Age-related diseases recede, and youthful vitality is restored. Notably, This transformation extends beyond epigenetic and metabolic aspects, encompassing external appearance, rendering us youthful not only in terms of genetics but also in outward appearance.